So welcome back, dear students. Let's continue with DNA fingerprinting. In the former session, we had spoken about the molecular basis about the differences which would contribute to fingerprinting. And this is an illustration of DNA fingerprinting, which has been used for the identification of a criminal at a crime scene. So now let's go into the details of fingerprinting, and this is Alex Jeffrey, the pioneer scientist who had put forward the process of genetic fingerprinting. Okay, and he was a man who was working mainly on the differences between DNA of different members of a family, and he noticed that in in the DNA of his technician's family itself, there was lots of difference. Okay, so he likely worked more on that, and he noticed that this differences which are being found mainly even in the members of the same family could be incorporated mainly to identify individuals. And the method which he developed then in the 1984s and all that mainly forms use in DNA fingerprinting over here. And now it is being used in the police detective work, and it is also proved useful in resolving various paternity as well as immigration disputes. So let's go into the details of that. So this is a man who is behind DNA fingerprinting. So what is DNA fingerprinting? In 1984, Alex Jeffrey, he is the one who discovered the DNA fingerprinting, and he realized that to detect the variations in the human DNA, you can use the mini satellites. The main technique, the technique DNA fingerprinting here mainly utilizes the differences in the mini satellites. DNA fingerprinting is a technique. It detects lots of mini satellites in the genomes to produce a pattern which has been unique to an individual. And this is a DNA fingerprint. And the probability of having two people with the same DNA fingerprint that are not identical twins is very small. If you are considering identical twins, they do have a chance of the same DNA, as well as the mini satellite pattern would be the same mostly. But in either cases, other than the identical twins, you can see that the chance of having a same mini satellite pattern or the mini satellites or a DNA fingerprint is being found to be very small. Just like your actual fingerprint, your DNA fingerprint is something you are born with, and it is very unique to yourself. So let's see what's it. How do you get this? To prepare a DNA fingerprint, what you do is you should of course get a DNA sample. Now this DNA sample could be anything. It could be your hair, it could be your bone, it could be your blood, it could be your skin cells, or it can be obtained from anywhere. Like if it might be any evidence which you get from a crime scene which could contain a DNA. So from that, basically, in whatever be the evidence, you extract DNA. This is not a DNA sample exactly, this is a blood sample which has been shown here. But since it is a source of DNA, they have written it as DNA sample. So the first step in DNA fingerprinting is to take your DNA and extract it. And after extracting it, that DNA is being cut by using restriction enzymes. What are restriction enzymes? These are enzymes which will cut the DNA using specific recognition sequences. And once you cut the DNA into small fragments, they are being cut, no, sorry, they are being separated from each other by using the process of electrophoresis based upon the size of now DNA. All the DNA has the same charge. It has a negative charge. But when you cut it, it gets spliced at different ends based upon the occurrence of the recognition sequence at different locations. So you will get fragments of different sizes, and when an electric field is being applied during the process of electrophoresis, the DNA fragments under the influence of electric field get separated based upon the size, because the charge is the same for all the fragments. And the shorter DNA fragments will be found towards the end, whereas the longer fragments will be found towards at the start itself. Not that they will not separate, they will get separated, but they would be moving at a slower pace. And after that, similar to the blotting technique which we had mentioned earlier, you will transfer this onto a particular membrane over here, and so the DNA fragments will get transferred to a particular membrane, and then you incubate them with labeled probes. After incubating them with labeled probes, you will what you will use a X-ray film, and what will happen, where all the probes are being attached, okay, you will get a DNA fingerprint. Now let's see what are all these. Now, steps into details. So the first one is to extract the DNA. Then using restriction enzymes, you have cut the DNA, and thousands of pieces of DNA with various lengths are being obtained. Then you do the process of gel electrophoresis, as I told you earlier. Based upon the size, they will get migrated, and it's under the what influence of electric field that this is being carried out over here. Now, after that, what happens? The fingerprinting step is being starting. You will extract the DNA from the sample. Sorry. Now, once the step has been sorted out, the pieces of DNA are being transferred. They've been blotted onto a particular membrane called the nylon membrane. And then what happens? You will get single-stranded DNAs onto the nylon membrane. And after that, it is being incubated with radioactive probes. Now, what are the radioactive probes over here? These radioactive probes will be specific to the mini satellite DNA, which is being tagged with radioactive phosphorus. That is, the probe which you use here would be matching towards those mini satellite DNA. You know, mini satellites are what? Repetitive elements, or they are satellitic DNA which are being found to be what? Repeated throughout the genome. So the chance of a probe to bind on to a DNA fragment is being found to be more. So where all these mini satellites are being present in the restricted digestive DNA, the mini satellite probe will go and bind. And it is also being tagged with radioactive phosphorus, and so you will develop it with X-ray film, and then you will visualize it. Okay, so when exposed to the radioactivity, a pattern of more than 30 dark bands appeared on the film where the labeled DNA was, and this pattern is referred to as the DNA fingerprint. And to compare two or more different fingerprints, the different DNA samples is run side by side in the electrophoresis gel. So now let me just show you into this. This is the what? This is a DNA fingerprint. So this is not in fact just, it's a X-ray film which contains the bands of what? The mini satellite probes have already been attached, and they were being developed. So after DNA isolation, you had digested it, moved it on an electrophoresis gel, and after electrophoresis gel, you have transferred those DNA onto a particular membrane, and that membrane was being probed using radioactive probes which are specific for the mini satellite DNA. And after the probes were being attached, they were being developed using an X-ray film, and every individual have a different, as I told you, the mini satellite based variations would be unique for each and every individual. So if you have three different suspects over here, the DNA of all these three suspects are being run in the same gel. Okay, and the DNA which you get from a crime scene is also being run on the same gel, and further you will get an X-ray film also which corresponding DNAs are on the same, and you compare the band pattern between the individuals and you can identify the suspects. So this is a case of DNA fingerprinting where you take a DNA sample, you cut it by restriction enzymes, and you get different fragments, do an electrophoresis, transfer it onto a membrane, probe it with labeled probes which are specific to the mini satellite. Why we use mini satellite? Because mini satellite is common to everyone, and but the number of repeats of these mini satellites will vary in different individuals. And then you develop it using an X-ray film, and there you get a fingerprint. So that's what about DNA fingerprinting. Hope so it is clear to you. Thank you for now.